Neurofibromatosis type 2 is a genetic disease that is characterized by vestibular schwannomas growing on the acoustic nerve of both ears. NF2 gene mutations within chromosome 22 result in the absence of the tumor suppressor myelin, causing the growth of these tumors. NF2 has multiple pathways, and I will be focusing on the RAC1-ROS pathway that occurs primarily within the cytoplasm. RAC1, CDC42, and ROS are activated by guanine nucleotide exchange factors, catalyzing the transfer of a phosphate group from GDP to GTP. Activated RAC1 moves to the cell membrane and stops contact inhibition, now allowing cells to grow despite contact. Together, RAC1 and CDC42 activate PAC1. This is seen as the first step in the cascade. In the next step, J&K is activated either by RAC1 directly, by RAC1 through PAC1, or by ROS. J&K in turn activates the transcription factor C-gen. C-gen then goes through dimerization with a CFROS protein to form the transcription factor AP1N, taking us into the nucleus. With CIF4E, which comes from a different pathway, AP1 causes the activation of cyclin D1. Cyclin D1 is a transcriptional co-regulator that interacts with AP1 to transcribe genes that cause cell cycle progression. When cyclin D1 is overexpressed, it promotes increased cell survival against antibodies and rapid cell cycle progression, resulting in the growth of the schwannomas. When the NF2 gene is not mutated, the presence of myelin stops the RAC1-ROS pathway. Myelin inhibits RAC1 activation by disrupting the GEFs, which in turn inhibits RAC1 from moving to the cell membrane to stop contact inhibition. CDC42 activation can't be inhibited by myelin, but myelin can stop RAC1 and CDC42 from activating PAC1 by binding to it. Myelin doesn't bind to ROS, but it stops ROS from activating J&K. Because no other activators of J&K are active, the pathway doesn't continue, resulting in the growth of no tumors. Thank you for watching!